Caitlin Martin, April 11th, 2023. This is my week 3 presentation for Genetics 1040. Bernardi's paper on the non-coding DNA mystery is a thought-provoking and enlightening piece of research that sheds light on one of the biggest mysteries in genetics. The central question of the paper is what the function of non-coding DNA is, which has been a long-standing mystery in the field of genomics. Bernardi proposes a new understanding of the genome that resolves this mystery, and his evidence suggests that the organization of non-coding DNA into isotopes is linked to chromatin structures. One of the most compelling arguments presented in the paper is Bernardi's explanation of the role of isotopes in shaping chromatin structures. Isotopes are DNA regions with different GC content, and Bernardi suggests that there are two superfamilies of isotopes that have different long-range 3D structures. He describes the GC-poor, gene-poor isotopes as low heterogeneity sequences with oligo-A spikes that mold the LADs, whereas GC-rich, gene-rich isotopes are characterized by single or multiple GC peaks that mold the TADs. Bernardi's evidence for this argument comes from analysis of DNA sequences, as well as data from other studies that have explored the relationship between the chromatin structure and DNA sequences. He proposes that the formation of these primary TADs may be followed by extrusion under the action of cohesion and CTCF, which highlights the importance of understanding the relationship between the chromatin structure and gene regulation. Another key argument presented in the paper is Bernardi's explanation of the genomic code. He argues that this code is responsible for encoding and molding primary chromatin domains, including LADs and TADs. This code resolves the long-standing problems of non-coding DNA, junk DNA, and selfish DNA by providing a new vision of the genome as shaped by DNA sequences. This has important implications for fields such as genetics, bioinformatics, and synthetic biology. Overall, Bernardi's paper represents a major breakthrough in our understanding of the genome and the role of non-coding DNA plays in gene regulation. By connecting the organization of non-coding DNA with chromatin structures, Bernardi has opened up new avenues for research in genetics and bioinformatics. His work provides a clear and concise explanation of isotopes, which can help researchers better understand how the genome works. This paper is a must-read for anyone interested in understanding the mysteries of the genome and the role of that non-coding DNA plays in gene regulation.